All right, everyone, welcome back. Here is our third and final lecture in Chapter 9, where we're learning about muscle tissue. After this chapter, I've said a few times, we go back to the nervous system, and that will wrap up our semester as we continue through nervous system. So, as always, let's go ahead and begin with our not really attendance questions. Just one today. How do neurons convey information for stimuli that are stronger or weaker? All right, hopefully you've paused the video and answered that question, or maybe you already knew the answer and didn't even have to pause, but let's see. Remember, an action potential always has the same amplitude. It goes up to 30 millivolts, positive 30, and back down. So if something is stronger or weaker, it's not that we get above or below 30. The action potential amplitude, it never changes. So what is it that means there is a stronger stimulus or a weaker stimulus? It was the frequency of those, how quickly those action potentials are coming. So instead of just one action potential followed by another action potential followed by another action potential, it's just one after another very, very quickly. So it's the frequency of action potentials that conveys strong or weak stimuli. So we're going to tie that in today with how we get stronger or weaker contractions. And there's a few ways that we do that. But before we talk about that, first we have to talk about what is a stronger or weaker contraction and what are the different types of contractions. So to do that, we have to learn some terminology. And the first term is tension. Tension is a word that we've all heard, but when we're applying it to muscles, it's going to mean something very specific. Muscle tension is the force exerted by the contraction of a muscle. So if you are lifting something that is 5 pounds, well, the tension within the muscle is something that has to overcome that 5 pounds as it is lifted. So tension is just the force exerted by the contraction of the muscle. And load is just the opposite. It's the force exerted by whatever we're trying to move with that muscle. So tension, the force exerted by the contraction of the muscle. Load, the force exerted by whatever we're trying to move with that muscle. And there are two different types of contractions: isotonic and isometric. Isotonic contractions, as tension within the muscle builds, we can see that down here in this graph, as tension within the muscle builds, the muscle shortens and moves the load. So that's what we're seeing here. We've got a load, looks like 3 kilograms attached to a muscle. As the muscle tension builds, the muscle shortens and we move the load. Now, isometric contractions, think about we're moving something or trying to move something that is way too heavy. You're trying to lift 500 pounds with one arm. All right, so as tension within the muscle builds, because it does still build, you can contract those muscles in your arms without them actually shortening and moving the load. So tension within the muscle builds, but the muscle does not shorten. Here we've got 6 kilograms, which is too much for this muscle to move. Tension within the muscle builds as it tries to contract, but it can't contract, it cannot shorten, so the load is not moved. Isotonic, tension builds, muscle shortens. Isometric, tension builds, muscle does not shorten. Okay, so now we're going to talk about something that we've probably already talked about in lab by this point, and that is motor units. The definition of a motor unit is one motor neuron and all of the muscle cells that it innervates. One muscle neuron and all of the muscle cells that it innervates. So try to picture this. Think back to what we were talking about on the first day of chapter 9. One nerve supplies one muscle. Nerve, remember, a nerve has a lot of neurons inside of it. And one muscle has a lot of muscle cells inside of it. But one nerve supplies one muscle. This muscle, we've opened it up so that we could see the muscle cells inside of it. And here we've opened up a nerve so that we could see some of the neurons inside of it. If we look at the purple one, labeled motor unit 1, we can see that as it comes out, it has endings, those axon terminals, on multiple myocytes, muscle cells. Same thing with the red one, motor unit 2. Here it has those nerve endings, axon terminals, on multiple different muscle cells. But they're all from the same nerve, just different neurons. Well, one motor unit is this purple neuron and these 2 muscle cells. Motor unit 2 is this red neuron and these 3 muscle cells. If only the purple neuron sends an action potential, only these 2 muscle cells will contract. If only the red neuron sends an action potential, only these 3 muscle cells will contract. If both motor unit 1 and motor unit 2 neurons send action potentials, all 5 of those muscle cells will contract. So even though one nerve supplies one muscle, we can get different reactions based on how many of those neurons send action potentials. So here's something else we kind of talked about in lab a little bit, and this is, how does a muscle cell respond to a single stimulus, a single bit of electrical activity from a neuron? And this is really, it's not something that happens in the body. This is a laboratory phenomenon when muscles are being studied. So we send a single stimulus to a muscle cell, and we get a single contraction. This is called a muscle twitch. And what we're looking at are the different phases of that twitch. The first bit is called the latent period. And what's happening during the latent period is the action potential arrives at the muscle cell. It travels, like we saw, along the surface, down the T-tubules. It causes calcium to be released. The cross bridges form, and that is the latent period. We see during the latent period, there is no increase in tension. Well, the next part is the period of contraction. This is when we are going through the cross bridge cycle. Tension is building as those sarcomeres shorten. And then, since we only got one stimulus, immediately all of that calcium is pumped back into the sarcoplasmic reticulum, the cross bridges break, and the sarcomeres go back to the resting state. So that is the period of relaxation. Tension drops. All of that is called a muscle twitch. It is the result of a single stimulus, and we go through that cross bridge cycle that we saw, but only briefly. The muscle does not noticeably shorten. So here that is kind of stretched out. Here we have a single twitch. Here's our stimulus applied, and then back to rest. But, like I said, that doesn't really happen. When we are sending signals to muscles in our body, we don't send a single stimulus. We are needing to do some sort of activity with that muscle. So what happens if we send more stimuli? Okay, in this image, we see the red indicates when the stimuli are sent. We have stimulus. We start to get a little bit of a contraction. We start to relax, but before we come all the way back to relaxation, we send another stimulus. So starting from that point, we get another bit of tension developing, start to relax before we come back down, another stimulus. So this is essentially wave summation. Remember wave summation from nerves? This is wave summation in a muscle. The result is something called incomplete tetanus. Tetanus is tension building within a muscle. So incomplete tetanus, we have a little bit of relaxation after each contraction period. And the reason that is because these stimuli are coming relatively slowly. But what happens if we increase the frequency of the stimulus? In other words, if we are having a stronger stimulus. That was why I asked that not really attendance question. If the stimuli are coming quicker because of a stronger stimulus on the nerve, well, here we go. We get contraction, but we do not get time to relax in between the stimuli. So maximum tension develops. It is very smooth. This is when you tighten your muscle. This is what you're seeing. This is called complete tetanus. Complete tetanus occurs when stimuli are so fast that there is no period of relaxation. We get increased tension, and the tension remains until the stimuli stop. Complete tetanus. Okay, so now we are going to start to look at motor unit recruitment. Remember, we said there are multiple motor units within a muscle. Even though it's only one nerve, that nerve breaks into multiple neurons, and those neurons serve different muscle cells. So let's see what we're looking at here in this middle portion, because this can be the part that's a little bit confusing. This is an imaginary cross-section of a muscle, and we are seeing individual muscle cells inside. When they are not colored in, these are relaxed muscles. The ones that we see colored in this sort of red color, those are the muscle cells that are contracting. So over on the top, we see stimulus number 1, not enough to reach threshold. Here we do not see any muscle cells begin to contract. So down here in the tension graph, there is no tension. Same with number 2. It's a little bit stronger of a stimulus, but it's not enough to reach threshold. So no muscle cells contract, no tension builds. Number 3, strong enough stimulus to reach threshold within the muscle. We get a couple of muscle cells contract. We get a little bit of tension developing. 4, 5, 6, 7, same thing. We're increasing the stimulus. We activate more and more and more and more motor units. So we have more of those muscle cells contracting. Tension is a little bit stronger each time. But here we can see in number 7, we've activated all of those muscle cells. So if we send a stronger stimulus, like we see with number 8, number 9, number 10, the stimulus keeps getting stronger, but in number 7, we already activated all of the muscle cells. You can't activate more than all. So in number 8, 9, and 10, even though the stimulus is stronger, we still have the same number of muscle cells contracting. So the result is in 7, 8, 9, and 10, the tension is the same. So what we're seeing is as we add stronger stimuli, we activate more motor units and tension builds. The strength of the contraction increases until we get to the maximum number of motor units. Once we get to that point, we can continue to add more stimulation, but we're not going to get more tension. Another thing that can increase tension strength is the size of the muscle fibers. Over here, the first motor units that are recruited, remember recruitment is just adding more motor units. When we start to add tension to a muscle, the first motor units are activating muscle cells that are pretty small. That's what we see here with motor unit 1. If we need to add more, if we increase the stimulus and we recruit more motor units, not only are we recruiting more, but they are bigger muscle fibers. And if we continue to add more, they're even bigger muscle fibers. So as we recruit, let's go back to our picture over here. As we are adding more motor units, not only are they increasing in number to get greater contractile strength, they are also increasing in size, which gives greater contractile strength. But what about if we're not stimulating muscle cells at all? Well, that's never really the case, like I said, unless you are either dead or paralyzed. If there is some sort of nerve damage or the nerves are dead, we are always sending a little bit of stimulation to our muscles, and that is called muscle tone or tonus. Tonus is the background level of stimulation to the nerves and muscles in our body. They are always sending a little bit of a signal. Now, it's not always to the same muscle cells. What's actually happening is the different motor units are kind of switching off. So we are constantly stimulating those muscle cells regularly, but they switch on and off. Even when you are at your most relaxed, you spend a day at the spa, you've gotten a nice massage, and now you're sitting in the sauna or something, whatever. When you are at your most relaxed, you are still stimulating your muscles at a low background level, and that is muscle tone. All right, so here we're talking about the sources of ATP for any sort of activity within our muscles. And there are different ways that we get ATP for our muscles. And the first is something called direct phosphorylation. Direct phosphorylation. And this is using something called creatine phosphate. Creatine phosphate. So creatine is a phosphate carrier that's found within our muscles. And the way that this works is creatine phosphate is going to directly transfer a molecule of phosphate to a molecule of ADP. And that ADP, when we add another phosphate to it, what does it become? ATP. We are left with something called creatinine. So this gives us one molecule of ATP. It doesn't require oxygen, so this can happen in anaerobic conditions, but it does not give us very much energy. We get about 15 seconds of energy from this. If we need more energy, we can go through that glycolysis bit of cellular respiration. Again, it does not require oxygen. We're using glucose. And from that, remember this first step of cellular respiration, we got 2 molecules of ATP per glucose. And since we're not using oxygen here, we also get lactic acid buildup. Lactic acid buildup causes a little bit of that burning in the muscles. Here we get a little bit more energy out of it, but still not much. We get about 30 to 40 seconds, maybe up to a minute. But then we have the aerobic pathway. We're going to start to breathe heavy. We're going to start to use oxygen. Here, this is that cellular respiration that we spent a whole chapter talking about. We get 32 molecules, roughly, of ATP per glucose. And we can get energy from this for a period of hours. So this is long-term energy. So how does this apply? Well, when we first start to exercise, if it is something short duration, like a sprint or something, the first 6 seconds, we're not really making ATP. We're using the ATP that's stored in the muscles. After that, for about 10 seconds, we're going to use that creatine phosphate. After that, we're going to start to use the anaerobic pathway. We're going to go through glycolysis and build up lactic acid. But if we're going to do long duration exercise, if we're going to run long distances, here we are using that aerobic pathway. We're using cellular respiration. So how do we get greater contractile strength? Well, lots of muscle fibers are recruited. Larger muscle fibers are recruited. And the sarcomeres, if remember from the previous lecture, if they're stretched just a little bit more. So let's look at that in a little bit more depth. We just kind of briefly mentioned it, but let's look at why that is. Slightly stretched muscle will result in greatest contractile strength. And up at the top, we've got unstretched muscle, unstretched sarcomeres, moderately stretched muscles or sarcomeres, and excessively stretched or overstretched muscles or sarcomeres. Which one will give the greatest contractile strength and why? Well, the slightly stretched will. Why is that? Here, when it's slightly stretched, look at these myosin heads and their relationship to the actin. Every one of these myosin heads can grasp the actin and form a cross bridge and pull that actin towards the M line. We get a lot of contractile strength there. If the muscle is not shortened, if it's not stretched, here we can see there's not really a lot of movement area. While we can still get a lot of cross bridges, they can't really do much because this muscle is already mostly shortened. Over here, if we overstretch the muscles, well, we cannot form very many cross bridges because most of these myosin heads, there's no actin around for them to grab. But right here, right in this sweet spot, slightly stretched muscle, that's where we get the number of cross bridges that are maximal, so that we can get full cross bridge formation and lots of room to shorten that muscle. All right, so that's it for skeletal muscle. Let's touch just a little bit on smooth muscle. We're not going to go into this very deep at all. First, we said smooth muscle is involuntary. We do not think about it moving. It happens as part of our autonomic nervous system. It is not striated. It does not use sarcomeres for contraction. Its shape is kind of ovoid. It's kind of an elongated oval with the ends that are pinched. It, instead of having neuromuscular junctions, it's got something called varicosities. Varicosities are where the autonomic neurons are going to connect to the muscles. If we look at autonomic neurons, instead of having those terminal boutons, they've got these swellings all along the end area of an axon. They act very similar, but the shape is very different. And right where they connect, there are things called caveoli. Caveoli are similar to those ligand-gated channels. And when an action potential is sent and the muscle is going to react, where does the calcium come from? Well, calcium is actually coming from outside the cell. Smooth muscle cells do not really have much of a sarcoplasmic reticulum, so they are not storing calcium inside. Calcium is coming in, so as this autonomic neuron sends an action potential to these muscle cells, it causes the opening of calcium channels, and those opening of the calcium channels at the calveoli, that allows calcium to come in. And then as calcium comes in, we have the action potential within the muscle cell. And the result is, here we can see kind of this web-like structure. That is actually where the tension develops. It's not really so much within the muscle, it's right on the inside of the plasma membrane of this muscle cell. As those contract, it squeezes the muscle in all directions. So the muscle cell kind of contracts in on itself. And instead of using troponin and tropomyosin, we use something called calmodulin. Calmodulin is what that calcium binds to, and it causes this movement, this contraction. But we don't have to attach to every single one of these muscle cells, because these muscle cells are joined together with gap junctions. So if we cause a bit of an action potential at one muscle cell, we can have that same thing occurring in the adjacent muscle cells because of those gap junctions. Here we've got cardiac muscle. Cardiac muscle is kind of similar to both skeletal and smooth muscle. It's involuntary like smooth muscle. It is striated like skeletal muscle. But unlike skeletal muscle, it's branched. It's not a straight, long fiber. It's a short, branched fiber. And the muscle cells join end to end with each other at the intercalated discs. At the intercalated discs, we have gap junctions and desmosomes. Desmosomes hold them together. Gap junctions allow that action potential to pass very, very efficiently between the muscle cells. Calcium, again, mostly comes from outside the cell. Here we can see that sarcoplasmic reticulum is not very extensive. So most of the calcium comes from outside the muscle cell. And when they contract end to end using that sliding filament theory like we saw in skeletal muscle. All right, there are a few disorders for you to know. We are not going to cover those in lecture. They're self-study. And that brings us to the end of the muscle system. Next time, we're going to be talking about the brain and central nervous system. We will have one more lecture after that, the brain. Be sure to use your outline for that because the chapter itself is very extensive. We are not going to go over everything that's in that chapter. We are going to abbreviate it quite a bit. So as you read, also watch the lecture and fill out your outline because most of that chapter we are not going to cover. Once we get the brain out of the way, we've got the peripheral nervous system basics, and that will be it. So we are very near the end of the semester, everyone. All right, take care. I hope you enjoyed this, and I will see you in the next lecture.